Okay, thank you for joining today's webinar. We at SoftGenetics are excited to release a new product to assist genetic researchers. My name is Kevin Levan, and I've been with the company since 2006. I've also been a part of many great advancements in genetic analysis. Next generation sequencing has aided the scientific community in a lot of ways, like drastically increasing throughput. But these technologies don't offer a complete solution. Some of the limitations of short read next generation sequencing include difficulty with aligning reads to the appropriate location of the genome, especially in regions like pseudogenes. It also struggles with reliably detecting many structural variations. Also, short reads don't completely overlap many significant repeat regions, and there are other limitations as well. Long read sequencing technologies help to advance research in these areas, but these instruments have some of their own limitations. For instance, accuracy, and also many of the available tools require a high amount of bioinformatics knowledge too. SoftGenetics has been a leader in the variant detection industry for over 20 years, first developing Mutation Surveyor, an aligner with patented technology for physical reference to sample Sanger sequencing comparison. Also has tools for low frequency variant detection and heterozygous indel deconvolution. We also have improved the accuracy and ease of use of other capillary electrophoresis techniques like MLPA and chimerism detection with products like GeneMarker and ChimerMarker. And when instrument manufacturers developed short read next generation sequencing techniques, SoftGenetics was ready with our NextGene software. SoftGenetics has been assisting researchers at the forefront of new sequencing technologies. So when PacBio developed single molecule real-time long read sequencing, and Oxford Nanopore developed nanopore sequencing, we wanted to make your analyses easier and more accurate. SoftGenetics is proud to release NextGENeLR, our software for long read sequence analysis. So for today's agenda, I will be discussing the following features in NextGENeLR. Its first release includes features for SNV and small indels detection, including features for minimizing false positives. Has features for structural variation detection to assist with identifying large deletions, insertions, and translocations. Several haplotyping tools are in NextGENeLR for targets like single genes or mitochondrial DNA and COVID-19. And lastly, features for identifying repeat expansions. Our goal at SoftGenetics continues to be a provider of software tools that are easy to use, highly accurate, and include visually informative results and reports, which we are going to go through today. If you have any questions, please type them into the chat, and if time permits, I will answer them at the end of the webinar, and we will also email you a response. So long-read sequencing can also be used to detect SNVs and small indels, similar to Sanger and NGS techniques. Doctors Stephen Mack and Henry Ehrlich of UC San Francisco have been using NextGENeLR for some of their non-invasive prenatal testing samples run on a nanopore MinION sequencing device. You can simply load a batch of FASTQ or BAM files into NextGENeLR, choose the reference you want to align to, and run. When it is complete, an output folder will have BAM and some VCF files and more. Click on a completed project and the pileup will open. The long read technologies currently have a higher error rate than the short read technologies. Read balance is one common metric used in short read NGS workflows to help distinguish true positives from noise. Because long read sequencing has higher error rates, SoftGenetics has taken an interest in improving that detection accuracy and sensitivity. Utilizing neighboring matches and mismatches can help to achieve this. So we've developed this effective percentage setting that is unique to NextGENeLR that does just that, helping to filter out lower quality variant calls. So right now we're looking at the NextGENeLR viewer. Light-colored nucleotides match the reference sequence in this pileup. Positions that are mismatched have a colored background. So gray background indicates mismatches that are getting filtered out, most likely because of low frequency. Orange background indicates positions where mismatches exceed minimum thresholds of detection, so the variant gets called. Many of these mismatches with a gray background are easily identified as small insertions. Alignment continues on both sides of the event without a problem. And because they are such low frequency, many of them are not called as variants. Some mismatched insertions and deletions cause a slippage of alignment because multiple possibilities are giving similar scores. That's why we see some regions with several consecutive bases that have a gray or orange background. So what can we do with these mismatches? Are they true positives? NextGENeLR has several solutions to minimize those false positives. At position 2242 of the HBB gene, over 7% of the reads contain a G nucleotide, and the variant table at the bottom shows that percentage. Upon visual inspection of the Gs with the orange background, we see that there are several neighboring mismatches in the reads. NextGENeLR uses this information in its effective count and effective percentage calculations. Basically, reads with more accurate local alignment are more reliable. This calls effective percentage is less than 1%, which can be seen in the variant table below. So I discussed varying percentages and coverages and effective percentage threshold, but there are other variant call quality metrics that can be used to help improve accuracy. These include allele scores, allele balance, and indel homopolymer length change too. So moving on to another feature in NextGENeLR, I'd like to show you our structural variation detection tools. Large insertions and deletions are common structural variations that can be detected with NextGENeLR. Several of the nanopore samples that UC San Francisco included a deletion that's more than 600 bases in length. This variant is known to reduce production of beta globin protein and cause beta thalassemia. And Sanger sequencing wouldn't be able to detect this with traditional methods, but CNV analyses could probably do that with NGS data. NextGENeLR is able to detect it utilizing its structural variation detection algorithm. And like the variant table, the structural variation report at the bottom includes a lot of useful information. We see information like the type of event. So here it's a deletion. We also see the position information, the breakpoints, depth of coverage, percentages, sizes, and more. So translocations have a tendency to occur at features like fragile zones or via non-homologous end joining or even single-strand annealing. And they're a good structural variation that NextGENeLR can detect as well. And here in this study from Rice University, some of the samples had undergone CRISPR editing and resulted in unintended large gene modifications. NextGENeLR was able to detect a translocation between chromosome 1 and 11 in this PacBio data. This image shows a portion of reads mapping to chromosome 11. You can see that this sequence is followed by a repetitive sequence. A lot of T's and some C's in there. In the upper right corner, I show the nucleotide sequence of one of the reads that aligns to this location. And you see that same repetitive sequence highlighted in blue. The lower pile up is showing the remainder of these reads aligning to chromosome 1. And you can see that they share that same repetitive sequence. The structural variation report at the bottom of the viewer goes into a little more detail and shows information about that event. So you can see here that the type is a translocation, and one of the breakpoints is in chromosome 1 and the other is in chromosome 11. This report has other useful information for helping to understand what's happening at this event too. And I do want to mention that structure variation report is output as a VCF file. So for haplotyping, I want to talk about the HBB gene. And phasing of SNVs and small indels can be demonstrated with short read NGS, but it's important. It's an important feature in NextGENeLR because we're also including structural variations with the phasing results. So to demonstrate the accuracy of the variant detection for the haplotype calculation, some of that UC San Francisco family data was used, where we have parents and siblings and a fetal sample as well. So there's a 2KB region of HBB gene that was sequenced using the Nanopore's MinION sequencing device from whole blood from the parental and sibling samples, and for the fetal sample is from chorionic villus sampling. For the haplotype report, the pileup gets split into three groups of reads. The groups on the left and center include reads that are organized into two detected haplotypes. So you can see haplotype 1 and haplotype 2 above these sections. And the group of reads on the right are those reads that don't match either of those detected haplotypes. Also in the pileup, the variants with a green background indicate positions used in the haplotype calculation that match the reference. The yellow background indicates variants used in the calculation that do not match the reference. NextGENeLR uses two calculations of linkage disequilibrium, and a third calculation also gets applied to each read. The first calculation is deviation, or D, and it's the difference between the observed and the expected frequency of one haplotype. The second calculation is R squared, or correlation between the pair of loci. In both of these calculations, 0 indicates complete linkage equilibrium, while larger numbers indicate linkage disequilibrium. And I mentioned the third calculation that gets applied to each read that generates a score for each haplotype. This calculation involves all variants within each read. Heterozygous variants are given a larger weight than homozygous variants, and structural variations have a weight related to their length to calculate the score. The first couple of lines of the haplotype report show the resultant haplotypes. So haplotype 1 on the left of the pileup is listed first in the report. Haplotype 2, the middle pileup, is listed second in the report. The remainder of the table shows an array of scores based on the deviation and the R-squared calculations for each position. Red background indicates positions with high disequilibrium, and so those are used to help identify the appropriate haplotype. NextGENeLR has included a pedigree linkage tool. So you can import the haplotype reports generated by NextGENeLR into this tool, and the first two columns in this case are representing the fetal chorionic villus sample. Columns 3 and 4 are from the haplotypes of the father. 5 and 6 are the haplotypes of the mother, and 7 and 8 are born and affected sibling. This affected sibling carries a 2 base pair deletion on the first allele listed near the bottom, and the 600 base pair deletion is on the second allele. So then you would set the known paternal and maternal haplotypes and click auto compare. This updates the haplo 1 and haplo 2 rows values and applies the color coding we see here to help with interpretation. The haplo rows, I guess the row shows the sample numbers that share the same haplotype. The first haplotype of the father's sample matches the corresponding haplotype of the fetal sample. And the mother's sample's first haplotype matches the haplotype 2 of the fetal sample. So this does not have that large deletion. Dr. Stephen Mack of UC San Francisco mentioned that NextGENeLR shows software makes haplotyping of the HBB gene accurate and easy for our Oxford Nanopore data. The software reliably detects large events, including the 619 base pair deletion. NextGENeLR is an essential tool in our long-read workflows. It was great to hear those positive words about a software, so thanks, Dr. Mack, and it was a pleasure working with you and your team. So let's look at some other haplotyping with some mitochondrial data. So you would load again your FASTQ files, choose the mitochondrial reference, and choose detect haplotypes and run. One important point to mention is that NextGENeLR will automatically display the amino acid translation relative to the appropriate mammalian mitochondrial genetic code. So when we zoom in on this region of ATP6 gene, we see a TGA that is translated to tryptophan. This would be a stop codon in the standard genetic code. And we also see an ATA that is translated to methionine, which would be an isoleucine in the standard genetic code. And because the mitochondrial genome is circular, it's important to note that NextGENeLR can align reads that span that origin. In the middle of the view here, we can see position 16561, and the next number we see is 29. So the origin is somewhere in the middle of that location, and we see that the reads are continuing through this without a problem. NextGENeLR has a built-in library for mitochondrial haplotyping. So for those of you interested in haplotyping, either of these, it's really easy. When we switch over to the haplotype report, we see the haplotype columns listed in the identified haplogroups as well. And you see major and minor for the mixtures too. So here we see that this sample has a mixture, let me zoom in a little bit more, of L3E and L0A haplogroups. The first is about 60% and the second is about 40%. Just like the mitochondrial haplotyping workflow, NextGENeLR can do the same with COVID-19 samples. So again, load their samples, choose the COVID-19 reference, and run. NextGENeLR includes a dictionary of known COVID-19 variants. Here is a COVID-19 project run on Nanopore's GridION system. The spike protein contains the position significant for haplotyping of this virus. And you can see the gene S up in that translation pane. You can also see codon numbers and reference and consensus sequences in the viewer. Spike proteins K417T is a key variant for identifying the gamma variant of COVID-19. The nucleotide track shows an A to C change. In the translation track shows the codon numbers as well as the lysine to threonine at 417. The variant table below the pileup also shows some important information. Like the variant percentage is almost 90%, coverage is 980, the amino acid change of again, K417 to T, and a lot more. So switching from the variant table to the haplotype report, we see that the haplotype report displays the subhaplotypes or lineages of COVID-19. This particular sample is a variant gamma B11. And we can also see the other consensus variants for this sample listed in this report. The viewer's haplotype report shows a single sample's results, but in most cases, these users will be running a whole batch of samples because these instruments can have very high throughput. So like the pedigree linkage tool is available for families, we have the global haplotype report for showing the results of large batches of samples. Here is a single table including all of the samples that were run on a COVID-19 project. This makes review much faster and easier. Here we can see the name of the sample, its haplotype call, a score of confidence, and the list of the variants. So we can see some that are alpha variants, some are theta, gamma, and delta. And when a batch of mitochondrial projects is added to the global haplotype report, we see a list of the sample names, the haplotypes, but instead of score, we see the major or minor contributors, indicating a possible heteroplasmy or multiple contributors. So it's definitely a very useful tool for mitochondrial sequences, COVID samples, as well as batch of gene haplotyping as well. But now I want to talk about STR expansion or repeat expansion. So because STRs are sometimes longer than short reads of NGS instruments, repeat expansion can be much more reliable with these long read sequences. NextGENeLR has a report to make this easy to do. I have a couple examples of repeat expansion. The first is from the University of Verona using the MinION sequencing device, publicly available on Sequence Reads archive. The second is from PacBio example datasets using their Vega benchtop system. So you would follow the normal steps of loading the samples, choosing the build 38 reference, and selecting to do detect structural variations. And the STR expansion detection algorithm uses the structural variation settings. Many of these repeats are quite long. And when the STR report is opened, a dialog opens listing some commonly researched STRs. So here we're looking at gene HTT or Huntington disease or Fragile X as another possibility. So this list can actually be edited too to include your own custom repeat expansions that you're analyzing. So once you click OK to get through the settings, the green bars, you'll get a plot that looks like this, listing all the positions that were evaluated. And then you see green bars that indicate the length of that's in the normal range. And then you see a gray bar for the region that's in pre-mutation range, and red when for regions that are in the full mutation or the affected range. So in this University of Verona, I mentioned that they use MinION, or maybe I forgot, they use MinION sequences to help characterize these samples. When we zoom in, we see that these Fragile X syndromes lines in the report show reads with 68 and 73 repeats. You can see that they're in the red zone, so that they would be unaffected. And the numbers in parentheses below that are the number of reads that have that number repeat length, so 18 and 63 respectively. Here is the example of repeat expansion using the PacBio's Vega system. And this is actually a pool of several samples. So one of the samples that has an STR in chromosome 4 and it's displaying reads aligning to the HTT gene. And these reads are clustering at the normal range with repeat lengths of 16 and 18. And the STR for gene PABPN1 on chromosome 14 has some reads with 10 repeats that are in the normal range, and some reads with 16 or 13 repeats, which possibly indicates OPMD or oculopharyngeal muscular dystrophy. And this disease is typically inherited as an autosomal dominant trait, so this is important to know. So just to summarize, NextGENeLR is a powerful tool for SNV detection, containing unique algorithms to help minimize false positives. It's a great tool for structural variation detection, being able to detect large deletions, insertions, as well as translocations. Has haplotyping features. NextGENeLR utilizes the SNPs or the SNVs as well as the structural variations for its haplotype calculation, which is a very useful feature. And it also has repeat expansion report designed to easily identify common as well as custom STRs. So NextGENeLR is just been released. We do have a 35-day trials available right through our website. And I want to thank you for listening to this webinar. You can get more information by contacting info@softgenetics.com, or you can just visit our website. Lots of information there. And I do have, I see a couple of questions that I want to answer. Um, so what are the system requirements? It's a very good question. So all of SoftGenetics products are Windows OS products. And for this particular product, we recommend using a computer that has at least 32 gig of RAM. For some of the bigger projects like Exome and things like that would need that. And the other question I see is can you can it align whole genome sequencing samples? And I do want to mention, no, not yet, not whole genome. I did mention it can do whole exome, but I'd say the majority of our users are doing small amplicons like single gene, or they're doing mito or COVID-19 for the haplotyping features, and then for STRs, yeah, like it could be whole exome for that. All right. Well, thank you again, and I appreciate you listening.